First of all, welcome everybody, which includes researchers, staff, students, especially students and staff. And welcome to JILAns and non-JILAns. Everyone is welcome. It's been more than 10 years since the last JILA Colloquium. I suspect many of you don't even remember, may have not been in JILA that long. If you're like me, you may have felt that you have gotten a bit out of touch with what is going on at JILA. You keep hearing about these amazing awards and endowed chairs and all kinds of things going on, and you have a dim notion that something excellent must be going on at JILA, but you're not quite sure what that is. So we thought it would be a good idea, especially as we, I hope, are coming to the end of COVID, to start a new series which is aimed at fostering collaboration and communication interdisciplinary across the board. And in particular, I hope that the speakers will make every attempt to make it understandable to me, because then it will also be understandable to you. I want you to share, Tom, I'm going to introduce you in just a second, but I want you to share the excitement of what you and your group is doing. This is not about you, it's about JILA and the excitement. On the subject of awards, Andreas Becker, who's the chair sitting up the back there, has suggested that this might be a good venue to acknowledge some recent awards, JILA awards to staff and students. So I'm going to read this, and please wait until the end, and then I'm going to ask the awardees to stand up briefly and we can give them a round of applause. JILA Jedi Awards for the first half of 2021 are awarded to, actually let me remove my mask so you can see my lips, Gail Geschwind, Lauren Mason, Tanya Rusi, Madison Foreman, Kenya Castleberry, and Andrea Martin for a demonstrated positive impact on JILA's culture of inclusivity and diversity, whether prolonged exemplary effort, leadership in impactful activities, or contributing to the inclusive and diverse culture of JILA in an extraordinary way. And then there's some awards, JILA Culture of Safety Awards, which are awarded to Kijong Yang, William Eckner, and Michael Hemmer, in recognition of their contributions to the review of JILA's Lazy Safety Consultant Report and development of recommendations thereof. Please, all of those who've been mentioned, stand up in your seats and take a round of applause if any of you are here. Or virtually, if you're on Zoom. Thank you so much. One more thing before I introduce our speaker, Tom Perkins, which is a sad thing, but something to celebrate as well. Dick McRae passed away 2 days ago. He was one of the first JILA astrophysicists back in the 1970s. Those who knew him remember a wonderful, sweet, generous person who was eternally agile in body and mind. He was my longtime squash partner. He usually beat me. Thank you, Dick, for all that you did for us and for JILA and for astrophysics. Thank you. You can do that. Now to our speaker. The plan is that Tom will take 40 minutes of talking, and then we encourage lots of questions. Tom, would you like to have questions during the talk, or would you prefer that they? Okay, let's say if you want to ask a question in real time, please raise your hand. If you're on Zoom, we're monitoring the chat, so you can raise a chat question, and Tyler is going to be looking after the chat. And if you think it's worth interrupting the colloquium, please let us know. Tom Perkins has been a fellow of JILA for 20 years. He's a professor in MCDB. He was chair of JILA 2018 and 2019, and he was acting division chief of the quantum physics division in NIST in 2019 and 2020. He has won many awards, and I have a short, never mind, I have dropped your awards. there it is, including the Governor's Award for High Impact Research, the Department of Commerce's Gold Medal, which is its highest award, and its Ron Brown Excellence in Innovation Award, and the Gears of Governance Award sponsored by the Office of Management and Budget. And there's more than those on top of that. Okay, there'll be snacks in the H bar following this talk. Yes? Okay, snacks outside afterwards. Thank you very much, Beth. Tom, take it away.
Okay, is my audio coming through? Good, excellent. Well, thanks to Andrew for restarting this series, and thanks to all of you for coming. Today, I'll be sharing the work that we've been doing where we've been developing techniques in atomic force microscopy to study the dynamics of biological molecules. And so, since this is really a general talk, let's start at the beginning, where we all know that the genetic information is stored in DNA, and in the process of transcription, that information is transcribed into mRNA, and then that is translated by what we call the ribosome into a linear chain of amino acids. But for a protein to have a function, it needs to fold. Now, that folding process is complex. Here's a molecular dynamics simulation of it. This is a very simple protein. It just has 3 alpha helices. The first thing that you'll notice about the process of protein folding is that it is bombarded by water molecules that's causing all of this Brownian motion. And then you'll see that there's states, that the protein gets stuck in a certain place for a little while. And so these states can accelerate protein folding, but they can also be traps. And then what you'll see is that this protein actually has to back out before it actually gets to its fully folded state. So what we really want to do is understand the process of protein folding. This is a 50-year-old problem in biology. And conceptually, what we think about is an energetic funnel going downhill. The fully folded state, or its native conformation, is often the lowest energy state. But to accelerate the process of protein folding, there are actually intermediates along the way. And there's not just one clear path. There are lots of different paths to go. Now, some of these intermediates are only transiently occupied. So if you're doing an ensemble measurement where you're measuring over trillions of molecules, if something only spends 10 microseconds here, you'd never be able to see it. And so one of the powers of the single molecule studies that I'll be talking about is that one can follow these dynamics. One can see an intermediate state that's transiently occupied. Now, it's not just the identification of the pathway in the intermediates. We're also very interested in the misfolding of proteins. Misfolding of proteins causes several severe diseases like Alzheimer's and Parkinson's. And so one of the powers of single molecule force spectroscopy is that you can actually map out this energy landscape. So if you're looking at small molecules as drug candidates, you can ask the question, well, does that small molecule stabilize the folded state? Does it destabilize the misfolded state? Or maybe it changes the barrier between the states. So we want to understand the process of protein folding. We want to be able to look at and measure the energy landscape. And one of the powers of single molecule force spectroscopy is that we can get a one-dimensional projection of this energy landscape. Now, there's a particular class of membrane proteins that drove us to use atomic force microscopy. They're ones called membrane proteins. Membrane proteins live in this phospholipid bilayer. On the top of this is a phosphate group. It's highly charged. It likes to interact with water. So there's water here. These little tails are carbon chains, so that's oil. And then on the other side, it's water here. So these phospholipid bilayers act as boundaries between the inside and the outside of the cell, or let's say the inside and the outside of mitochondria. Now, the blue things are membrane proteins. Membrane proteins represent about 30% of proteins in the genome, but approximately only 1% of the protein data bank. However, they're critical to many processes of life. If you want to transport ions or small molecules across this lipid bilayer, that's done by membrane proteins. A lot of signaling in cells are done by membrane proteins. Membrane proteins can also act as catalysts, and they're also integral to the structural aspects of the cell. Now, as such, membrane proteins are very important biomedically. They represent 50% of current and future drugs. They're also very interesting scientifically. Insights associated with structures of membrane proteins have been associated with 4 Nobel Prizes in chemistry. Now, what we'd like to do in studying these membrane proteins is sort of look at a fold. So let me rephrase that. There's a lot of membrane proteins that fold into a particular type of fold that has 7 transmembrane helices. So these are these transmembrane helices going from one side to the other. This family of proteins that adopt this include a very important class of membrane proteins called GPCRs, or G protein coupled receptors. Biomedically, these are important. 35% of all drugs target GPCRs, and there are approximately 900 to 1,000 GPCRs in the human genome. Now, we'd like to understand the energetics that drive this protein folding process. But membrane proteins live at this oil, water, and highly charged interface. So understanding the energetics is very complex. But energetics can also lead us to understand the conformational dynamics of these proteins, look at ligand-drug interactions, and understand the origins of disease-causing mutations. So how do people measure the energetics of membrane proteins today? Or traditionally, what you do is you have something diagrammatically here, which would be the native fold of a membrane protein. You would extract that membrane protein out of its native lipid bilayer and put it into a detergent micelle. And then what you do is you increase the amount of detergent, what's called SDS, and that denatures the protein. It unfolds the protein, as shown here. And now one of the problems, as I just mentioned, is you can't do this in the native lipid bilayers, but the energetics are dictated by the protein interacting with those lipids. So that's one downside with this technique. Another problem is that, here is a measure of absorbance as a function of mole fraction. So this is the experiment that you do. You measure an absorbance of a chromophore in this case, and it goes down as you increase the mole fraction, and then it unfolds, and then you end up in an unfolded state. Now, one of the problems of that unfolded state is it's not really unfolded. As diagrammed here, about 70% of the alpha helical content of that protein is still there. So it's thermodynamically not a particularly well-defined state. What you've really done is you've broken up a lot of the tertiary interactions between these alpha helices, but you actually haven't unfolded it. Another problem is that it's very difficult to make this process reversible. There are only a few membrane proteins where you can unfold something and refold it, and therefore it's very difficult to get at the underlying energetics. And then there are a number of assumptions involved in analyzing data like this that the literature has raised, not the least of which is it assumes a 2-state model, and I'll show you that there are a lot more than 2 states in the unfolding process. So what are we going to use? We're going to use a tool called single molecule force spectroscopy. Here is a diagram of an optical tweezers type of assay just to get you grounded in single molecule force spectroscopy. Many of you are familiar with optical tweezers. Here we're pulling on a RNA hairpin. And interestingly, when you pull on this at constant force, it exhibits 2 states, folded, unfolded, folded, unfolded. So even though it's 25 base pairs long, you only see these 2 states. So this, of course, is a cartoon. This shows you the high resolution data that we can get with these types of experiments. And so with this type of high quality data, of course, you can measure the unfolding rate and the refolding rate quite easily. But underlying those parameters is something that I discussed earlier, which is the energy landscape. So we'd like to not only know the kinetics, but we'd like to learn aspects of the energy landscape. So this is energy in this direction. This is the so-called nebulous reaction coordinate. When you pull on something in a single molecule force spectroscopy experiment, that reaction coordinate becomes the direction on which you're applying force. Now, force times distance is energy. So when you apply a constant force, what you do is you tilt this energy landscape, and you take a situation which would normally be only occasionally thermally occupied, and you get to the point where it's occupied 50-50 between unfolded and folded, just like you see here. And so this is what we measure. And if we do a good job, we can measure parameters of the energy landscape or even the whole energy landscape itself, and then reverse the process and go back and get the native properties that the biochemists will be interested in. So the question that we've been exploring for about 15 years is, can we study membrane proteins with a similar level of control as what our colleagues have worked out to study small RNA hairpins and structured RNA. Okay, so this is the underlying assay that we're going to start with. This is an atomic force microscope cantilever that you see bending. And what you're going to do is press it into a lipid bilayer that contains the protein. This protein called bacteriorhodopsin has 7 transmembrane helices. And what you see is that pairs of helices come out at one time. Okay, so the primary intermediate that we're going to see is pairs of helices coming along, and that's what was established in this original paper. And here is a force versus extension plot. Whenever the force goes down, something unfolded. Okay. So I'm going to talk a fair amount about AFM cantilevers. So just to give you a sense of what an AFM cantilever looks like, there's a chip. This is generally on the scale of a few millimeters. It's something that you can pick up with your hands. But here is a set of SEM images, scanning electron microscopy images, that show you what a cantilever really looks like and the business end of the tip, which is sharp on the scale of a few nanometers. So that's what a cantilever, AFM cantilever looks like. Now, one of the powers of atomic force microscopy is that you can take atomic or near atomic scale pictures of things, and you can do that in liquid and biological molecules. So what you see here is a picture of bacteriorhodopsin and its native lipid bilayer. It forms a 2-dimensional crystal. You've probably seen bacteriorhodopsin if you've flown into San Francisco and you're going over the salt ponds and you see this crazy purplish chemical stuff and you say, what's that pollution down there? That actually what you're seeing is this molecule here. It's a light harvesting complex in archaea that grow in high salt ponds. And so it's called purple membrane. This is the dominant color that you'll see in those salt ponds outside of San Jose. Now, in the original experiment, they took these beautiful high resolution images. Now, to initiate the assay, what you do is you take an AFM cantilever and you just push it into the surface really hard, a few nanonewtons on our scale. And then about 1% of the time, you get a record that looks like this. You got lucky, you attached to what's called the C-terminal tail of the membrane protein, and you pulled the whole thing out. Now, you could make arguments from polymer physics that say, hey, I'm pulling on one molecule, but the power of being able to image is that these pioneering experiments, they went back and imaged the lattice and show that, yes, I picked up just one molecule. Now, what we want to do is we want to be able to do these equilibrium assays so we can understand energetics and better. But now here we have a force versus extension plot for the type of things that we get. So now let me walk you through this. So if you're going up along this line here, which we'll call 88, it means you're pulling on 88 amino acids that are unfolded. So structurally, you're in this state where you're pulling on the top of a pair of helices, the so-called ED helix pair. You've already pulled a pair of helices out. And so if you follow the red curve, there's a force drop. That means something unfolded. And what happens is this whole E helix unfolds and you go to this state over here, which has 105 unfolded amino acids. And then you can go up and then the force drops and you pulled out the whole D helix. Okay, now each time you go through this process, it's thermally activated, so it's a little bit different. So sometimes you populate an intermediate along the unfolding pathway. Now, when you're pulling on these experiments relatively fast, it's a thermally activated process. And so the faster you pull, the higher the force is. So most experiments are done up in this regime, so it's pretty easy to distinguish between these states. But if we want to do an equilibrium experiment where we pull more slowly, the problem that we're going to run into is that the distance between these lines, which tells us which one of these 3 states that we're in, is comparable to the noise. So even if we can see something, we can't tell you which one of these 3 states it's in, then a biochemist won't care. Okay, so what we've really had to do from a metrological perspective is work on improving the precision, the temporal resolution, and the stability of bio-AFM. Okay, so what I'm going to do is tell you that how we did this. To give you a sense, we're going to get down to a temporal resolution of about 1 microsecond in liquid. So this is the mechanical response time of one of these cantilevers. And that's going to enable us to do these equilibrium studies that we want to do. And here you can see beautiful near equilibrium data back and forth between 3 states. Importantly, we can assign these states structurally. This top state is the fully folded bacteriorhodopsin, and here are where the next two states are. To give you a sense of scale, the distance between these three states corresponds to a mere 8 amino acids, are the unfolding of 2 turns of a single alpha helix. So these are distance scales are very small. And here you see is 10 milliseconds. The dynamics are very, very fast. Okay, so what I'm going to do now is tell you a little bit about how we make these cantilevers. And then we're going to transition into talking about how these modified cantilevers dramatically improved our ability to see dynamics of molecules that were previously hidden. And I will conclude by showing how what we've done generalized to a number of other biological systems. Okay, so we've made a set of modified cantilevers to improve performance. We wanted to improve the stability. The first thing that we found out is that if we strip the gold off a cantilever, that we improved the stability by about a factor of 50. These cantilevers were relatively long and soft, and so in liquid, they had a mechanical response time of about half a millisecond. And then we went and we started using something called a focused ion beam that I'll tell you a little bit about in a minute, which allowed us to get to modify shorter cantilevers. This cantilever is just 40 microns long, that now have a time resolution of about 70 microseconds. And then we went down to cantilevers that are really tiny, just 9 microns long. And in liquid, these cantilevers have a mechanical response time of just a single microsecond. Okay, so how did we get to this point? Well, the problem to start with was that the force stability of bio-AFM was historically quite poor compared to optical tweezers. And we did an experiment where we just take the cantilever and watched it for a couple minutes. We loaded it into an AFM, we sit it there, it's in liquid, let it sit there for a few hours and then measure it. And what we saw is that the 0 position of the cantilever drifted by tens of nanometers over minutes. Okay, this is really horrifying because that corresponds to a force change on the scale of nanonewtons, and we're trying to measure things on the scale of piconewtons. Now, we knew it was the cantilever was drifting because we could bounce the laser beam off the back of the cantilever and so that was quiescent. So yes, the problem is not the stability between the tip and the sample, which is what people always assumed. It's actually the cantilever itself. So Allison in our lab developed the ability to measure this position of this cantilever, not just over minutes, but over hours. And we could see that it's drifting by hundreds of nanometers over the timescales of hours. But what she found was that if we stripped the gold off the cantilever and the underlying chrome, that this became really stable. So it's the metallic coating that people have put on AFM cantilevers that have really been the source of the stability problem. So diagrammatically, if you have a metal-coated cantilever, you get more light off of it, but actually the drift in that cantilever is 50 times worse. Okay. So now, as we try to further improve things, let me talk you through a little bit about the rationale about how we think about this. So what we're going to do is we're going to take a cantilever and just measure its thermal motion in liquid. And here is one of these long, soft cantilevers without gold, so it's much more stable than a typical cantilever. And so we take the thermal noise spectrum and we transform it, and here we're going to look at piconewtons. We're going to look at force because molecules respond to force, not position, and as a function of frequency. And at low frequencies, we have instrumental drift. Many of us who are experimentalists are well aware, everything would be ideal, but something goes wrong on long time scales. This is that something wrong. Now, in an intermediate regime, we're thermally limited. So unlike our colleagues who study quantum resonators, the Q for these types of things are 0.5 or a little bit less. So we're in the over-damped or just perhaps critically damped regime. And then on faster timescales, you can't see things because the cantilever can't respond fast enough. You've got a diving board in liquid, and there are things that you just can't see. So, we think of this as sort of the response time as the cantilever, what we would call the roll-off frequency. Now, the mantra in these types of experiments is smaller is always better. Go to a shorter cantilever, life will get better. And why is that? Well, physics will tell us that the force noise in a certain bandwidth is governed by this equation, just the square root of 4 kT times the bandwidth of the measurement times the hydrodynamic drag of the cantilever. Now, as someone who has to do experiments at room temperature or a little bit above room temperature, the only thing as an experimentalist that I can change here is the hydrodynamic drag of the cantilever. So smaller cantilevers, better. Now, what you see here when you look at these smaller cantilevers that everyone would say is better, yes, the noise went down a little bit at higher frequencies. The response time went up some. This is good. But the primary thing you probably see is that the low frequency noise of these cantilevers actually got dramatically worse. So better on timescales faster than 30 milliseconds, but on longer timescales, they actually perform worse. But we took an important lesson from this data, which was we thought, well, softer cantilevers give us stability, smaller cantilevers give time resolution. So what we wanted to make was a soft and short cantilever. But the problem is that the stiffness of a cantilever goes as 1 over length cubed. So if we want to go to a shorter cantilever, the problem is it's going to be stiffer. And that's really where Matt Bull, who was an undergraduate in the lab, stepped up, and he created these focused ion beam modified cantilevers. So this is really focused ion beam modification is really, think of it as sandblasting with atoms. And so he made a cantilever like this. And this cantilever is really special because it has a little gold patch to retain high reflectivity, but got rid of gold everywhere else. So particularly down here where there's curvature in the cantilever, that's really where the stability of the bad aspects of gold happen. And then he cut a hole in it and he thinned it. And so it's tenfold reduction in hydrodynamic drag and a tenfold reduction in stiffness. So how did he do this? We put down a little mask of transparent material to protect the gold. And then he cut the cantilever and then he thinned these supports. And then he did a simple etch where we removed the chrome and removed the underlying gold. And this dramatically improved the performance of the experiment. So here's a very common assay in our types of things. You take a bunch of protein domains and you hook them up head to toe and you attach it to a cantilever in a surface and you pull, okay? And this is actually not, this type of head to toe linking of protein domains is actually present in your muscle. The largest protein in your body is something called titin, and it's got lots of things that look exactly like this. So when you pull on a structure that looks like this, you get a force versus extension plot where it looks sawtooth. And every time the force drops, one of these protein domains unfold. So this is where we took this cantilever that Allison developed, which was state of the art at the time, and then we looked at the same system with one of these cantilevers, and what you can see is that the data improved. And just we can zoom in and can see we went from force fluctuations on the scales of 10 piconewtons to now just a few piconewtons. So that was a real improvement in our ability to do the measurements. So we had done this with cantilevers that were 40 microns long. And so now the idea was, can we do this, take this technology and shift it down to cantilevers that are just 9 microns long? And the answer is yes. And the critical thing here was an insight provided by another undergrad, Javen, who realized that if you look at this process with an electron microscope while you're modifying, you can solve one of the big problems that we had. One of our problems was the cantilever bends. So these silicon nitride cantilevers want to bend up. And we need the cantilever to be straight. So when a laser beam comes in, it bounces off to our detector, whereas if it's up like this, it's going to bounce off and we're not going to be able to measure it. So we need the cantilever to be straight. And so what Javen developed was the ability to watch the modification of the cantilever while we're modifying it with a focused ion beam. Okay, so he's making a series of cuts, it's bending, it's getting worse and worse. But what he found was while he was thinning it, it straightened out. And so then if he stopped the process, the thinning process, when the cantilever was straight, we went from a yield of about 1 in 20 to almost 100%. And this was a really critical insight for making this process usable, these cantilevers usable on a day-to-day basis. Okay, so I've told you about these cantilevers that we've developed, and now we want to apply these cantilevers to biophysical systems. We're going to use this cantilever over here. These are the ones with 1 microsecond resolution to give you a sense of scale. The performance of these cantilevers correspond to 100 times higher time resolution and 10 times higher force precision than previous studies of membrane proteins. So you've seen this force extension plot before, and we're pulling on a pair of transmembrane helices, and there are 2 intermediates that you see, which correspond to these 2 states in the force extension curve. Okay, so now when we apply these cantilevers, we first thing that we see is the data is just dramatically better. Okay, and this is because the cantilevers have higher performance. Now this particular unfolding curve has one intermediate. Now, when we did it another time, it's a thermally activated process, we get a different unfolding pathway. And we do it another time, and we get yet a different unfolding pathway. And we can do it again and more times, and we get different unfolding pathways. So what we learned here is that their prior studies over about 15 years said that there were 2 intermediates when unfolding a pair of transmembrane helices. Now with these modified cantilevers, we see at least 14. Or put differently, prior studies were missing 85% of the unfolding intermediates. Now, one of the things that we noticed in these records is that the force goes down and comes back up, the force goes down and comes back up in every single record that we saw. What this means is that local parts of the protein are unfolding and refolding, unfolding and refolding. And this was really unexpected because we're pulling at a relatively high speed here. Okay, and almost certainly what's happening here is that these proteins have always been unfolding and refolding, but we just didn't have the time resolution and the sensitivity to see them. So it's of dynamics and intermediate states uncovered. Now what we want to do is we want to take these series of states that we've learned on these force extension plots and then translate them to amino acids that our colleagues who are biochemists care about. Because, you know, as a force spectroscopist, this gets me excited. But what biochemists want to know are what are the structural intermediates along the pathway? So how do we do that? Well, we have a force extension plot, and we have elastic models called a worm-like chain model that allows us to measure, let's say, the distance between these 2 states. In this case, it's 0.7 nanometers. We know the sort of structural properties of unfolded amino acids, and we know the projection of the alpha helix along the stretching axis. And if we take these parameters in a simple model, we can find out that this corresponds to the unfolding of just 3 amino acids. Okay, so it's really remarkable that you can see something with such high signal to noise unfold, refold in real time. And importantly, these bacteria rhodopsin molecules are in their native lipid bilayers. Okay, so now we can take curves like this and translate them into a number of amino acids, which means we can assign them to structural states. You know, the dynamics can be very complex. So here we have a force versus time plot, and this looks a beautiful trace where it's going from the 3rd to 4th to 5th state back and forth. And it looks like here you go from the 5th state directly to the 3rd state. Now I mentioned the time scale here is just 1 millisecond. So these are time scales are really fast by single molecule studies. But with these cantilevers that we have, you can zoom in and you can see that when you're going from the 5th state to the 3rd state, that you spent 7 microseconds in this intermediate state. And then you bop down to this state for 12 microseconds and you went back up. So the dynamics, there are states that are being populated on the shortest timescales, and there are even things that are too short for us to call with any sort of statistical confidence at the moment. So, you know, we can call things down to about 7 or 8 microseconds. For people who are chemists and think about these things as you go from one state to the other, okay, you go across the energy barrier. That's called a transition path time. So these timescales of sort of 1 to 10 microseconds are really what people have associated with transition paths between states rather than the states themselves. Okay, so we've talked about being able to identify transiently occupied states. We'd like to understand unfolding pathways. So here is a force versus extension curve, which is a particularly simple one. Over here are a set of diagrams. So this is the fully folded, this is an intermediate, and this is after you've pulled that pair of transmembrane helices out. And so the unfolding pathway here is quite straightforward. Now we can take a different record and we can see that its unfolding pathway is more complex. It has refolding in, and a number of different states are populated. So we can look at a different individual trajectory in its unfolding pathway, and we see yet a different unfolding pathway. And then we can take a collection of these and begin to map out this intricate network that was much more complicated than we ever anticipated. There's folding, there's unfolding. So there's a very rich system to explore. Okay, so we've seen intermediates, we've seen unfolding pathways. Now we want to get to energetics. And one of my biochemistry colleagues said, you know, Tom, all that beautiful data that you just showed me was all after a pair of transmembrane helices had been taken out. I don't really care about the energetics of membrane proteins after you take out a pair of transmembrane helices because it's no longer in its native fold. And so the problem is that, you know, this part of the unfolding curve is really easy to study. But right down here, there's a lot of tip sample adhesion. And the reason that is you take a cantilever that doesn't have any chemistry on it, and you push into something really, really hard and hope that it sticks. Okay. And so we took on this challenge and developed a chemistry where we put a particular chemistry moiety called an azide on a PEG-coated tip. The PEG is important because it's a non-stick agent. And then we mutated the bacteriorhodopsin so that we could put a particular chemistry called DBCO on it. And for my chemistry colleagues, DBCO is a copper-free click chemistry reagent. But the important point here is that under a microscope, you can form a covalent bond between the azide and the DBCO. So you can covalently attach the tip to the molecule that you're pulling on, and you can do so at a site-specific location. And when you did that, all of a sudden, this initial part of the unfolding becomes much cleaner. It becomes much cleaner, so now we can actually analyze the data. The first thing that we see is that there's actually a state here that was previously undescribed, and I'll talk to you about that in a minute, but there's some sort of fuzz here, and if we zoom in on that fuzz, we see beautiful near equilibrium dynamics, unfolding and refolding between 3 states on very short timescales. Here, it's 2 milliseconds. You've seen these 3 states before. These are the ones that we can assign. This is the fully folded bacteriorhodopsin. As I said, this is unfolding of 8 amino acids. And this unfolding of 8 amino acids puts this bottom state right where the photoactive chromophore, the retinol, is. Okay, this is the light absorbing compound in bacteriorhodopsin, and it stabilizes this bottom state. Indeed, what we found was this previously undescribed state is the single most mechanically stable state in the whole unfolding pathway. Okay, this covalent attachment allows us to do experiments over longer timescales. Okay, so now we can do beautiful equilibrium types of measurements where we watch it, let's say, flicker between 2 states, and then we tilt the energy landscape a little bit more, and we can bias it in being whichever one of these states that we want. And now if you get a record like this one shown here in blue, where it's bouncing back and forth between 3 states, you can measure the probability distribution of being in those 3 states as a function of position. And why is that important? Well, if we measure the probability as a function of position with high enough time resolution, we can actually apply an inverse Boltzmann analysis. Okay, we can derive the underlying free energy landscape as just minus KT times log of the probability distribution. There's some important corrections that I won't go into here, but this allows us to get at the underlying energy landscapes now for membrane proteins starting from their fully folded state. Now, this is a perturbed energy landscape. But now if you get rid of that force, you can see that it's energetically downhill for these membrane proteins to fold. Okay. So now we want to start looking at the energetics of point mutations. So here is the helix, the first helix to unfold, the so-called G helix. These 3 filled black circles are the amino acids corresponding to the fully folded state and the first two intermediates. And here's a force versus time plot. And David Jacobson in the lab went and engineered in point mutations into the system and measured their change. And what you can see is that the dynamics change as you mutate these amino acids. So what's beautiful here is that you can see the dynamics change for a single point mutation. Okay, so this is force versus time, but we want to get to energetics. Okay, so what David did is he developed a beautiful assay along with another postdoc, former postdoc Hao Yu, where he used a non-equilibrium measurement to measure equilibrium energetics. So what they're going to do is unfold, refold, unfold, refold, unfold, refold many times, and then identify lots and lots of transitions going up and down between these states. These 3 states are coupled, so it's not state 0 to 1. If you're in state 1, you can go either to 2 or back to state 0. So when you're deducing the transition rates between these, you have to be a little bit careful. But as expected, the unfolding transitions from 1 to 2 increase as you increase the force down here on the bottom axis, but the transition rates decrease as you increase the force for folding transitions. And there's a special force which we call F1/2. And if you could do the experiment exactly at F1/2, you would have an equal probability of being each of the 2 states. So once you identify F1/2 for these types of transitions, you can think of this notionally as a force extension plot, where F1/2 is sort of this unique thing where you can calculate the work as the area under the curve. Okay, you have to make some corrections, but then you can get the free energy of unfolding this set of amino acids. And so that's delta G, that's the unfolding free energy. Okay, and that's associated with unfolding a number of amino acids. And there are two different transitions here. But what we want to do is use this energy that we're measuring now to start probing the energetic consequences of single amino acids to the overall free energy that stabilizes these membrane proteins. Okay, and we're so here, David first looked at leucine 223. So this is an amino acid that faces inward. So this is the rest of Bacteriorhodopsin. The blue helix is the G helix, and the magenta is the particular amino acid that we're looking at. And what people typically do is change those amino acids to a small amino acid called alanine, and that tells you, well, what was the energetic consequence of this? And so interestingly, the AFM measurements and the prior types of measurements done before in ensemble biochemical assays using SDS essentially get the same change in free energy associated with just that single amino acid. But that's good. But what was interesting is when David looked at this valine 217, the free energy change associated with that particular amino acid was much, much larger in our AFM assay than prior SDS experiments. Now, interestingly, this amino acid faces outwards. Now, this had us confused a little bit, but David inspected the structure and realized that there is a structurally resolved squalene right here. So that means the protein is touching a lipid strong enough to stabilize it. And so if you get rid of that interaction, it has energetic consequences, which helps explain why there's a big change in the free energy measured by SDS and what we see when we measure it in native lipid bilayers. Since we want to understand the process of protein folding, we'd like to understand it in the native lipid bilayers. So what we think we've done is developed a platform where we can measure the energetics of membrane proteins in their native lipid bilayers. Okay, and now in the last few minutes, I want to tell you some other systems we're taking this technology and applying it to. Okay, so I've told you a lot about membrane proteins, but they're standard globular proteins. And then within those, there are very simple proteins like a 3-helix bundle. This is sort of a computationally derived protein. It's very small. And we can take this type of technology and try to understand things about it. So this particular protein, alpha 3D, is interesting because prior studies had suggested that the energy landscape at neutral pH, shown here in black, was rough, whereas the landscape had the same barrier height, but was smooth if you lowered the pH. And so what Devin in the lab did was he developed the ability to measure the equilibrium folding and unfolding of this tiny protein that unfolds at relatively low force. And he could measure it at a particular pH, and then we can use hidden Markov models and deduce the unfolding and refolding statistics. And then what he saw that is indeed, as you lowered the pH, that in fact, the kinetics of folding and unfolding quickened. And so this is just sort of an illustration that we can now use these AFM measurements to measure a wide variety of proteins, including ones that unfold at very low force. But then we got interested in saying, well, can we apply this technology to things that are more biomedically relevant? And luckily, Marcelo Sosa, who's a biochemist here at CU, came to us with a project where he wanted to study a class of proteins called effector proteins. Okay, effector proteins go through something that's called the type 3 secretion system. And basically, the bacteria takes a protein and pumps it into a host cell, our cells, human cells, to sort of make it more virulent. Okay, so gram-negative bacteria tend to use this. So many of you will be familiar with Salmonella. So that's one of the types of human pathogens that use this strategy. Now, these effector proteins here are two called SPTP and SOP2, where the crystal structure is known, go through this tiny protein needle. The protein needle is just 2 nanometers wide. So you may ask, well, Tom, where does all this unfolding and refolding of protein come into play for biomedically relevant systems? Well, it's certainly true here that if the protein exists here and ends up here, it has to unfold and refold to go through the process. Okay, now interestingly, if you put other proteins, one called green fluorescent protein among and attach it to an effector protein, the whole system stalls. It doesn't work. So GFP can't go through it, whereas these proteins can. And the underlying paradigm for many years was that these types of proteins are thermodynamically unstable. They're just on the edge of stability, and that's what lets them get through. Marc-Andre in the lab showed using standard techniques that, in fact, these two proteins have very typical thermodynamic stabilities, and indeed indistinguishable from green fluorescent protein. On the other hand, he took AFM data that showed these two proteins unfold at very low force, whereas green fluorescent protein unfolds at high force. So what this has done is given us a new paradigm for these systems, that it's not the thermodynamic stability, it's how easy for these proteins to unfold that governs whether or not these effector proteins can transit from a bacteria into a human cell. Okay, and to generalize off of proteins, there are short RNA hairpins shown here. I showed you an assay at the beginning where we're using optical tweezers to probe this. And typically those are done in specialized rooms. We have one right over here, but now we can do these experiments with AFM cantilevers. So we can get the precision, the time resolution, the stability to do these really challenging experiments, and we can even reconstruct the underlying energy landscape. Seen here is sort of the asymmetric energy landscape that is very typical for unfolding a hairpin. So what I've told you in this talk is how we've used advances to study the energy landscape for protein unfolding and refolding in a field that's called single molecule force spectroscopy. But AFM is used broadly in biomedical science as well as nanotechnology. And so the advances that we've made in the stability, the time resolution, and the force precision should be immediately broadly applicable to lots and lots of other biological systems. Okay, so to summarize, we've developed cantilevers where we can do single molecule studies with 1 microsecond time resolution on a commercial AFM. These cantilevers have revealed a multitude of hidden intermediates that were previously unobserved. We can reconstruct energy landscapes from equilibrium or non-equilibrium types of data. We've determined the energetics of membrane proteins. Particularly important is we've done it for membrane proteins in their native lipid bilayer. And I've shown you how we can generalize from just studying membrane proteins to a multitude of types of systems. And with that, I would like to thank my group members, current and past collaborators, and you for your attention.
Thanks very much, Tom, for an amazingly clear talk. So if you have any questions, please raise your hands. Tom, maybe you could take the questions rather than...
Yes, I got it. Okay. We could try to so let me repeat the question for Zoom. So if I get it right, you want to say, can we anchor the membrane protein to a surface, grab the membrane protein or a lipid?
Grab the head group and then just pull it so you straighten it out, so you unfold it, and keep applying force strictly.
Okay, so the question is essentially, how much force does it take to break a peptide bond? So yes, you could covalently anchor a peptide to a surface, and you could covalently anchor a tip, and then you could pull on it, and then you could ask, how much force does it take to break one of those bonds? It gets tricky because the peptide bonds are going to be breaking at about the same force as some of the chemistry where we anchor the protein to the surface or we anchor the protein to the tip. So the question is, which bond breaks? So we've shown we can get up to like 2 nanonewtons. The Gaub lab, which is a lab in Germany, has gotten up to like 2.5 or 3 nanonewtons worth of force. And the problem is at that level of force, you're really wondering which bond breaks. Okay, because if you're going to use a silane chemistry to attach to a surface or a gold thiol bond to attach to a surface, likely gold thiol bonds will probably break before the peptide bond.
So when each different way that break bonds, like have different active quasi-stationary states, so states that are long-lived but not necessarily stable, like would you be figure out what intermediate pathway you move on.
Okay. Okay. So I guess, okay. So this is a question of language. So when we think of these intermediate states, okay, so a chemist would think of a bond as something that's covalent between these intermediates are a network of like hydrophobic interactions, salt bridges, entropy that stabilizes intermediate states. So these intermediate states in the unfolding pathway are not structurally bonded together, but it's a whole network of energetics that cause a particular intermediate to be in a state. Did that clarify?
Yeah.
Okay. Over here.
Yeah. What essentially comes out of this data a lot is the information about the secondary structure. So what about systems that exhibit really strong tertiary or quaternary structures? Can you get information from like what breaks out first if two alpha helices are interacting with each other? If you just break out, will those bonds break out first, or will it be the alpha helix breaking out?
So it depends on the system that you're applying it to. So let's say, so generally the answer when you pull with force, you're applying force along a vector, okay? And what unfolds first depends on what that vector force is. So if I take a shoelace and I tie it and I pull on the loops, nothing's going to change. I keep pulling, and nothing happens. Okay, so clearly the direction of force can matter because I can pull the ends of the loop and it just comes undone. So when you pull on something, you can bias which thing unfolds. Now you can break a protein-protein interface. So if you have two things, you can pull on them, and sometimes the interface will break. Or sometimes an alternative is that you start to, let's say an alpha helix unwinds that weakens the interface. So how do you distinguish between those can become complicated. And that's you can do some clever experiments to try to understand that. You can do molecular dynamic simulations to try to understand, because of course we just see force versus time. There are other types of experiments where you can say, oh, I think this is unfolding first, and so I'm going to put in a salt bridge there and stabilize it, or I'm going to put in a metal-metal bond and stabilize it. And then if it gets stabilized, yes, I know that in fact was the weak link. And so there are a number of different experiments you can do to try to infer what the order of unfolding is. And you can probe protein-protein interactions. We've done an experiment where you take a protein and you put a ligand in and you pull a ligand out of a protein, and you can measure the energetics of that. So there are a whole variety of different types of systems you can do. The key is trying to understand how well can you interpret that data to make a conclusion.
Just following up on that, so bacterial synthesis does not show a lot of protein alpha helix interactions.
No, there are a lot. There are a fair amount of interactions. I mean, in that SDS experiment that I showed you, most of the energetics that people have probed historically have actually been tertiary interactions, because when you put this thing in SDS, what you're really doing is you're not getting rid of the alpha helices, you're just breaking the tertiary interactions between the helices. Okay, and that's what all the classic membrane protein energetics have studied. They actually haven't unfolded the protein. There's 70% of the alpha helical content is still there.
Got some questions.
Oh, okay.
No, you're good actually.
Okay.
We're watching for
Okay, back in this colorful shirt.
I'm wondering about the gold content of problems. Is that something like the gold is spongy, or having it's a thermal drift with like two different
Let me, okay. I have a slide for that because I get it asked. Yep, thank you. So the question is, what causes gold to induce drift? Okay, so one theory would have been, or was, that gold is in the silicon nitride forms a bimetallic strip. Okay, these are great thermometers. Okay, well, these types of cantilevers are actually coated on gold on both sides. But gold is actually viscoelastic. So what we think is going on, and what you see here in this scanning electron micrograph, is you take these cantilevers that are incredibly thin and long, and you pass them through a water-air interface, and they fold back on themselves. Okay, and then Matt Bull image the damage in the gold coating caused by going through this air-water interface in the gold coating. So we think there's stress that's induced into the gold coating in the process of wetting it, okay? Gold is also known to be viscoelastic. So it turns out people who do LIGO found out that metallic coatings of gold were bad, and that was actually the clue to us that gold might be a problem here. So it was actually stuff out of LIGO that gave us a hint that gold might actually be the underlying problem.
Maybe relevant for the previous question, but for membrane, studying membrane, the motivation is the forming how it is folded. Then you only search for the landscape of this direction. This, like this particular fluid direction, energy landscape relevant to the folding process. How this experiment contributes to the of the different folding.
So the question is, was the motivation of the bacteria rhodopsin experiments probing the energetics of membrane proteins? And that was absolutely our original motivation. We wanted to develop a set of tools, and bacteria rhodopsin, I didn't say, is the model membrane protein of choice. That's sort of, it's what everyone uses. But what we're doing now is we're moving from just studying the energetics of bacteria rhodopsin to being able to study its function. It's a light-driven proton pump. So we're interested in studying that. And what we want to do is take the technologies that we've developed here and start applying it to other membrane protein systems that may be more biomedically relevant. But bacteria rhodopsin is a very robust, simple system to start with. And so it's sort of,
My question is, you only study the 1D landscape.
yes.
But I think there are many ways energy landscape is more.
Absolutely.
How much
Okay, so the question is, we measure a one-dimensional energy landscape, and how well can we measure energetics, and to what degree does the higher order of the landscapes, so these landscapes are, are huge multidimensional things, and what we're doing is we're projecting this tremendously high multi-order landscape onto one axis because that's what we can measure. And there are certainly other axes where things are happening that we're not seeing. Now, with respect to determining energetics for delta delta G, we're just fine, okay? Because if you're measuring the change in free energy from a well-defined reference state, well, we can measure the change, the energetic consequence of that. I mean, it turns out the free energy of unfolding by pulling is about 20 times higher than the free energy change by doing SDS denaturation, because the SDS denaturation actually doesn't get rid of lots of helical content. It doesn't, and so the energetics we're fine with. But you're right that when we think about membrane protein folding, and one thing I didn't talk about is that membrane proteins fold into their lowest energy state, but inside our cell, there's machinery that stitches it in. And so the process of protein folding for membranes in a cell is driven by a molecular machine called a translocon, but it folds into its lowest state. And so there's thought to be different processes in the folding where these alpha helices may be sitting on a lipid bilayer and then go down into the lipid bilayer and then associate. So when we're measuring forces by pulling an alpha helix this way, we're not measuring the forces that, let's say, compress helices together to form tertiary interaction, but we can measure the energetics. So it depends on, you're right that it's a higher order system, and we're going to always get a one-dimensional projection, but there's a lot to learn from that, but not everything.
Just following up on that question, the energy landscape is usually very high. Dimensionality of energy, to some extent, because when you define a free energy, you average over degrees of freedom that you have decided are not relevant. And so you're always reducing the dimensionality. It's a matter of, are you reducing it too much or are you not reducing it too much? So I was just noticing that when you change the pH, the energy landscape, free energy landscape became much more rough. And I was wondering, is that a signature, the appearance of roughness in your derived landscape is a signature that there's another degree of freedom in the problem that's now becoming important? Now, because motion in that degree of freedom is being kind of projected onto your one dimension and making it look messy.
Okay, so if I repeat the question,
Can I interrupt at this
Yeah.
point? Because we started on time and it's now 1 o'clock and I'd like to start a tradition of ending on time. So if you'd like to remain and continue these questions, by all means, go ahead. But let's give Tom one more round of applause. Thank you very much, Tom. Those of you who want to stay behind, you're welcome to continue asking those questions.